I'm Michelle Vierra. I head up our strategic marketing team for plant and animal sciences. And after we saw all those really great talks about how smart sequencing was used, I want to spend a few minutes just to chat with you about how far we've come in the past year and the things you can expect over the coming months. So Jonas showed this slide a little bit earlier, and I love it because it really does demonstrate just how far we've come over just a few years. But for me, 2019 was a really special year, so I want to focus on that top right corner and talk about a little bit about what we've done in that year. In fact, it was exactly 1 year today that a preprint went up on BioArchive that described highly accurate long reads, or HiFi reads, to improve variant detection and de novo assembly of a human genome. And in this paper, they showed that you can get high precision and recall of all types of variants, whether it be single nucleotide variants, indels, or large structural variants, all across the human genome. They also showed you're able to phase these variants into their individual haplotypes, which led them to detecting 5% more variants in the medical exome, or specific genes that are relevant to human health and disease. Furthermore, they actually assembled this highly accurate long-read data, and not only did they get really high contiguity, but they also got very high accuracy. So for me, this was the first demonstration that there was a difference to using this highly accurate long-read data and made it really powerful for studying genomes. And then just a few months after that, we had the release of the Sequel II system, which, amongst other things, made it possible to generate 8 times more of this highly accurate long-read data so that it was easily accessible to do these large-scale projects. And as shown in the quote at the bottom of the screen by Luke Talon, who runs the core facility at the University of Maryland, the Sequel II right out of the box was pretty much production ready. So we saw a huge adoption of generating large amounts of this HiFi data. But naturally, we're PacBio. It didn't stop there. A few months after that, we released the 2.0 chemistry on the Sequel II system, which improved sequencing even further. And we did this by increasing the efficiency of these reads going into a rolling circle synthesis and continuing all the way out to the end of the movie, which really improved our average read length up to 25 to 50% longer, and overall gave you more yield of these HiFi reads by up to 40%. But what was interesting to me is by increasing the read length, you could create these HiFi libraries that went up to 25 KB in length. And I went, aha, I work in plant and animal sciences. Once you get these highly accurate long reads out to 25 KB, you're being able to now tackle some of these complex repeats that we see in plants and animals. And so it was these 3 developments to me that really said, hey, look at all of these applications we can do in 1 to 2 smart cells on the Sequel II system. That means we can start tackling large pan-genome projects, we can multiplex into tons of things for targeted sequencing, and makes it really efficient. And speaking of applications, we had a lot of updates on these individual applications. So taking a look at whole genome sequencing, we get a lot of questions from the whole workflow, starting at sample prep all the way down through bioinformatics. I think it's safe to say anybody at PacBio, the #1 question we get is, how do I extract DNA for my particular organism for my genome project? After looking at this for several years now, no surprise, there is no one-size-fits-all answer, but there are thousands of publications that have come out of people doing genome projects, so clearly the information is out there. And so Jonas, bless his heart, taking it upon himself to scour the literature and pull papers that had great methods for DNA extraction across a wide variety of organisms, put together this very easy to scroll, yet not totally phylogenetically accurate tree, to where you can go find your organism of interest, click on the paper. So for instance, going to the fish section, clicking on the 3rd paper, you are linked directly to the methods for how they extracted DNA from the yellow belly pufferfish for the genome project that they did. So we hope this will be a really easy to use tool for you. It's extractdna for pacbio.com. You can scroll, see if there's something useful for you as a good starting point. If you can't find what you need there, or if you want to include a protocol that maybe you have for an organism that's not represented, please get in touch with us by emailing extractdna@pacb.com, and we'd be happy to help you or incorporate your workflow. Moving on to library prep, I think the #1 change we saw in 2019 was that HiFi became the new standard for genome projects. And that's because the resulting assemblies were really high quality And by high quality, I mean thinking about what I call the 3 C's, which is contiguity, completeness, and the correctness or the accuracy. And so with everybody wanting to do their genomes with HiFi data, it became absolutely necessary to create a simple library prep to work for all HiFi workflows. And we achieved this by adapting the Express Template Prep Kit 2.0 to be able to generate HiFi libraries. In 2019, we also saw the release of the low DNA input workflow, where you're able to generate high-quality genome assemblies from small insects from only about 100 nanograms of DNA. And we've already updated that a couple times to work with HiFi workflow. And with the incredible throughput of the Sequel II system, you're now able to multiplex at least 2 different 300 megabase genomes in a single SMRT Cell 8M. And if you want to know the best way to tackle your specific organism of interest to create a reference quality genome so that every species can be a model, I encourage you to stop by Sarah Kingan's poster number 235 to see all the different ways that you can do that. We know that 100 nanograms isn't always possible to get from every organism, and that a lot of the tree of life is dominated by really tiny organisms. So we started working on the ultra-low DNA input workflow. This requires only about 5 nanograms of input DNA. Can probably go lower, but we're early on in development there. It also takes advantage of the HiFi workflow so that you use these highly accurate long reads. It's a simple all-in-one kit that you can buy directly from us. It's amplification-based. You just amplify your DNA, make a SMRTbell library like you would a normal HiFi library, and sequence away. We're currently in early access with this, so if this is something you're interested in trying on a particular organism you're having trouble with, please get in touch. We'd be happy to collaborate. But so far, on the organisms we've tested it on, it's worked really well. So here's an example of a collaboration we've had with a few folks at Notre Dame and UC Davis to sequence the Sandfly, which is the image in the top right corner. It's very, very tiny. You can only get about 20 nanograms of DNA out of one of these things. So starting with only 5 nanograms from a single Sandfly, we amplified it, we made a SMRTbell library, and then we sequenced it on a single SMRT Cell 8M, yielding almost 36 gigs of HiFi data that actually had a mean read quality of Q36, so that's pretty incredible. And not surprisingly, the assembly came together beautifully. The genome came together at about 363 megabases. We solidly got into the 1 megabase contig club, and we had 97% of the BUSCO genes that were complete. This did have 100-fold coverage from a single SMRT Cell 8M, which just points to the fact that multiplexing is going to continue to be an important priority for quickly being able to sequence lots of things. Jonas actually has a poster, number 129, that goes through the full details of the project. So if that's something you're interested in, again, go talk to Jonas. The poster session is right after this workshop, and he'll be able to tell you all about it. And if you thought, hey, 5 nanograms of DNA, that might be really important for human sequencing in thinking about, say, tiny needle biopsies or things like that, you're not alone. We did consider that as well. So we've started developing the application for variant calling across the human genome using the ultra-low HiFi workflow. So far, we've tested and gotten really good precision and recall in both SNPs and IDDELs across the whole human genome. But I will give the caveat that this is very early work, and we are currently working with Google to train deep variant for this particular data type. And we will continue to work towards improvements across the whole workflow for this in the future. Continuing on the analysis side of things, we saw lots of updates in 2019 here as well, starting with the instrument control software 8.0, which had an improved base caller. Always good. We always want more access to more high-quality data. And then we also improved the CCS algorithm so that it sped up significantly. So the time to compute your HiFi reads decreased by 5 to 7-fold. Our software assembly team, including Zev, worked really hard to adapt Falcon and Falcon and Zip to create these dedicated pipelines to take in HiFi data that's optimized for both sequence and phasing accuracy, and taking advantage of newer tools like PBMM2 for speed. I think we're not even fully understanding how good HiFi assemblies are, but we are trying to think of new ways to assess the quality of these genomes. And BUSCO isn't always the answer, but we did dive into that a little bit Sarah Kingan has a poster number 110 that can show you just how high quality some of these genomes are and how far you need to dig to find errors in them at all. And I think it's worth spending a little bit of time. You saw, I think, hinted at it in Bart's talk, that when using HiFi data to assemble a genome, you use significantly less compute. And that's because the file sizes are smaller and the assembly process is shorter. So here's a table comparing the de novo assembly generation of a human genome with both HiFi reads and long reads. And what you see is that the file size alone is about 1/7 of the size for HiFi, and all in analysis time, including CCS analysis, read correction, and contig assembly, takes about half the amount of time as it does with long reads when you're using the kind of PacBio recommended compute infrastructure. And we aren't the only ones who are noticing this. In fact, Kevin Fengler is giving a talk tomorrow during the Corteva workshop where he's describing how you generate a maze reference genome assembly in a day. And I think he was being conservative with the day, because last time I talked to him, it was down to a few hours. So I highly encourage you to check that out. That's tomorrow afternoon in the Corteva workshop. So just kind of to reiterate, whole genome sequencing on the Sequel II system is looking really good. Most projects can be done in a single SMRT cell or two, and the analysis time is really quick. I like to joke that 10 out of 10 bioinformaticians choose HiFi over long read data because it's so much easier to work with. So taking a look at RNA sequencing as an application, I think most everybody here is probably familiar with what Iso-Seq is, but just as a quick refresher, Iso-Seq is PacBio's full-length cDNA sequencing solution. And because it's full length, that means you don't have to assemble transcripts on their own. You can do it in a targeted or whole transcriptome fashion. And what we see when people do Iso-Seq projects is that they oftentimes discover novel genes and isoforms, even in really well-characterized samples. And this really allows you to improve your genome annotation, whether or not you have a reference genome. It's also used a lot to increase the accuracy of your RNA-seq quantification data down to the isoform level. It's also a really, like Zev said, a really easy application to do in kind of 3 days' time, a 1-day library prep, 1 SMRT cell project with a full suite of bioinformatics solutions depending on what kind of questions you want to answer. So zooming in on that bottom part a little bit, it was back in June that we released the IsoSikh Express protocol for faster RNA sequencing. It contained an RT-PCR kit and a new IsoSikh sample prep protocol that allowed you to go from total RNA to a sequencing library in a single day, while also reducing the input requirement down to 300 nanograms of total RNA, which ultimately reduced the cost by about 1/3. And with the throughput we're seeing on the Sequel II system, it only takes 1 SMRT cell 8M to do really any project. So here's just an example where we're showing you can comprehensively characterize a whole human transcriptome in a single SMRT cell 8M. And like I said, there's lots of tools to answer a variety of different questions. So Liz created this tool called IsoPhase that allows you to explore allele-specific expression in your sample. There's methods like DartSeq that allow you to look at phase 6-methyl-A detection in these transcripts. And we see a lot of projects in plant and animal sciences that are actually presented at PAG. One example is this Brahman-Angus hybrid cattle, where they used Iso-Seq to look at both allele and tissue-specific isoform expression. There was a recent cannabis preprint that just came out that used Iso-Seq to look at tissue-specific transcripts that are associated with both THC and CBD synthesis. And they also used it to annotate the Y chromosome. And if you followed our SMART grant at all, or joined us for our North America UGM, you might know that we're using Iso-Seq to look at grizzly bears, studying tissue-specific alternative splicing in hibernation versus active state, to look at insulin resistance, which has implications for type 2 diabetes research. And besides the question of how do I get DNA out, I think the other question we get most often is how many SMRT cells does it take to do my project? And when it comes to Iso-Seq, the answer is always 1 SMRT cell. And that's because you do hit saturation on whole human transcriptome in a single SMRT cell. And if you're wanting to do, say, a genome annotation project and get a wide variety of transcripts to look at, you can actually multiplex. Here it says 8 tissues, but you can actually multiplex up to 12 tissues in a single SMRT cell. In 2019, we also saw an uptick in the variety of targeted sequencing projects we saw taking advantage of SMRT sequencing. One really cool one is the Herbal Consortium, which is a Moore Foundation-supported 7-year project to barcode tens of millions of samples to catalog the biodiversity of the world in a slightly different way than the EBP. They actually got their Sequel II system. They can barcode up to 40,000 species in a single SMRT cell. So in 2020 alone, they're going to do species identification on millions of different samples. And this is a cool heat map showing one Sequel II SMRT cell where they multiplex 36,000 different species there. So that's an immense level of barcoding that we've never seen before with SMRT sequencing. Not surprisingly, we also see a lot of targeted projects looking at resistance genes across different plants and animals, some in tomato or Arabidopsis, a cool paper in Czech indigenous cattle that we see. And an area that kind of piqued my interest was biomonitoring of herbal supplements. So when you think about taking a capsule of herbal supplements that has a bunch of different stated herbs in different quantities, how do you know that you're actually getting what's supposed to be in there? And there were some really cool papers out of Asia where they barcoded and tracked what supplements were in there so that they matched the expected biological composition, which is really cool. And it's safe to say that, again, this is another affordable option because you can barcode a crazy amount into a single SMRT cell. So I don't think it's important to look at the numbers there because that number can be basically whatever you want. Lastly, we've had a lot of updates on our microbial sequencing side of things as well. So when you think about sequencing individual microbes for genome assemblies, naturally the Sequel II system makes it lower cost because you are able to do higher multiplexing. We have 48 different validated barcoded adapters, but what's the coolest thing about this application is we have a dedicated microbial assembly pipeline that allows you to go from loading your sample on the instrument to a single contig assembly that's circularized around the origin of replication, ready to submit to NCBI with the single push of a button, which makes it really easy to do this in a high throughput manner. HiFi reads are also really useful in profiling metagenomic samples. One method of doing that is looking at full-length 16S sequences. A couple different ways you can do that. You can do it with self-ordered primers in a PacBio one-step protocol, or you can actually get a kit from our partner Shoreline Biome that allows you to extract, amplify, and has the tools for analysis all in one. And the importance of looking at full-length 16S versus looking at just subregions is you get rid of the bias in what subregions are able to classify as far as taxa go. So this is a recent paper out of the Weinstock lab where on the figure on the right shows different subregions that were used to classify a database sequences that they knew. And the more red you see is the more sequences that they were not able to classify. So red is bad, white is good. And so even with really commonly used subregions like V4, there's a whole lot of sequences that they are not able to classify. But in contrast, in the top right corner, the V1 through V9, which is the full-length 16S region, you're able to classify almost all of them. That's not really surprising, but as I say in the quote here, sequencing the entire 16S gene provides real and significant advantages over sequencing commonly targeted variable regions. Another way to look at your metagenomic sample is via shotgun sequencing with HiFi, and this gets you 10 to 15 kb DNA fragments that are these high-quality HiFi reads, which means you're actually getting multiple genes per fragment, which allows you to do direct functional analysis on the reads without assembly. And so this is a table of 9 different human fecal samples that we sequenced on the Sequel II system with the HiFi protocol. And the main thing is to look at the bottom here. So we get an average of over 2 million HiFi reads, so 99% accurate reads in every single SMRT cell. And the average read length of these individual reads is almost 9 kb. And so when you think about doing, say, a metagenomic project with short reads, oftentimes you're using assembly to be able to do your gene prediction. And if you look, these read quality metrics are better than most contig assembly quality metrics for short reads. So what does that read length and accuracy do? Like I said, it allows you to do gene discovery directly on the HiFi reads. So here's another 5 different human fecal samples that we sequenced. with an average number of predicted genes of about 19 million. Each gene is around a KB on average, and you're getting about 8 genes per HiFi read. So even without assembly, you're getting really, really important, useful information out of every single read in your data set. It's also a fast, easy, and affordable application on the Sequel II system because of being able to multiplex many samples, whether it's individual microbes or your metagenomic samples. And so I think overall, it's safe to say 2019 was a pretty bang-up year for smart sequencing, and we saw that in the uptake and adoption from all of you guys, and we appreciate that. But the year is now 2020, so what have we got going in the future? Looking ahead, on the system and sequencing side of things, I think it's safe to say you can expect a chemistry or two release from us so that we can increase throughput and read length and be able to sequence one human-sized genome via HiFi on a single SMRT Cell 8M. We also want to be able to push through the number of genomes per year we can do on a Sequel II system, so we're looking to reduce the movie time for generating HiFi data so that it's faster. We're also still working hard to improve CCS analysis, both in terms of speed and accuracy. And eventually, what we'd really like to have is CCS analysis directly on the Sequel II system so that what you get right off of a sequencing run is your HiFi reads as a FASTA or FASTQ file. In thinking about the end-to-end applications that we like to develop, as throughputs continue to go up, naturally we need to increase multiplexing across all of our applications, whether it be Iso-Seq, targeted sequencing, low input, et cetera. And you can look forward to the release of our ultra-low DNA input kit and probably additional developments on that front. We're also looking to implement UMIs for quantitative Iso-Seq development. In fact, yeah, that'll be something that we'll be looking to collaborate with the community on. We're also wanting to release a single-cell Iso-Seq protocol and accompanying support. Actually, Liz Sang at the last minute has a talk tonight in the single-cell workshop that you can pick her brain on how things are going as far as single-cell Iso-Seq goes. On the software side of things, we are looking to be able to implement HiFi reads as direct input for HGAP4 or assembly directly in the SMRT-Link GUI. As more tetraploid roses and other genomes are coming about, we want to have improvements and tools ready to tackle diploid and even polyploid genomes as far as assembly goes. And as part of that, we want to improve phasing by using the inherent data built in to the HiFi data type. So for example, asking questions like, instead of phasing reads before assembly, as in trio binning, or phasing it after assembly, as in unzip, can we actually phase during assembly? And in fact, Zev and his team are working hard on answering this question already and developing some tools. So if you're around on Wednesday, I'd love for you to come by and hear from Zev on that and contribute to that discussion. We're also always wanting to improve our workflow support for those who work directly with the instrument and the tools. So things like deploying SMRT-Link into the cloud or streaming data directly from the instrument to the cloud, providing support for demultiplexing many, many thousands of samples directly in the SMRT-Link GUI, developing tools around base modification and additional graphs for that. But the honest truth is we have tons of ideas of what those next bullets should be, but ultimately we want to hear from you, our users and our advocates, to know what things we should be working on and tackling in the future. So please get in touch with us. Open lines of communication. We love hearing from you. And the last thing I want to say is I hope this is not our only interaction with you here at PAG. We do have 4 amazing posters. I only touched on these topics a little bit today, but please stop by these posters, talk to the presenters, check into the workflows and the research a little deeper. And if you haven't stopped by, we do actually have a booth here on the exhibit floor. It's booth number 511. We are giving away free sequencing, which is really fun. We're doing this in collaboration with our 5 different certified service providers who are also exhibiting here. All you have to do is fill out a small form, tell us what kind of project you want to do, and you'll be entered to win. There are 5 chances to win, and this is not that big of a conference, so I think it's worth entering. We also brought back our limited edition trading cards, our PacBio, our Pokemon-style trading cards. They are different from the ones last year, so definitely stop by the booth, ask us how you can catch them all. And then we are also hosting our annual Smart Informatics Developers Conference on Wednesday afternoon here at the Town and Country. You do not need to pre-register, you can online, but we'd be more than happy for you to stop by and join in on the conversation. It's an afternoon of short talks and a whole lot of discussion about where tooling for smart sequencing is headed, what problems we're encountering, how we're solving them, and I will put the caveat that you do not have to be a developer to get something out of that conference. I go every year, and I find it to be a really great afternoon of discussion. So with that, I'll turn it back over to Jonas and say thank you for sticking around.